In late 2019, numerous cases of severe viral pneumonia were admitted at the hospitals of Wuhan, East China. The following months, the number of similar cases increased geometrically around the world. On February 12, 2020, the disease was named Coronavirus Disease 2019 and referred to as COVID-19 since then. On March 11, 2020, the World Health Organization officially declared COVID-19 as pandemic. Its full name is Severe Acute Respiratory Syndrome Coronavirus Type 2 or SARS-CoV-2. There is an entire family of coronaviruses. Most of its members cause symptoms of the common cold. However, MERS-CoV, SARS-CoV, and the new SARS-CoV-2 may cause severe respiratory failure in humans. The new coronavirus consists of three main parts: its spikes, allowing the virus to attach and invade our cells, an envelope that protects the virus genetic material, a genetic material essential for its replication. In nature, two types of genetic material exist: DNA and RNA. Depending on the presence of specific enzymes in its organism, a DNA molecule can replicate itself. A DNA molecule can be transcribed to RNA. An RNA molecule can replicate itself or can be reversibly transcribed to DNA. All derived copies contain identical information with the parent molecule. Animals, humans, as well as some viruses contain DNA. In humans, our DNA is located in the cell nucleus, forming our chromosomes. Using a DNA template, fragments of DNA named genes generate RNA transcripts. They are called messenger RNAs or mRNAs. mRNA molecules exit the nucleus, are translated into proteins, and they are dissolved. Coronaviruses contain RNA. Every piece of coronavirus RNA represents an actual mRNA containing information for the production of its components, and it's ready to be translated. Coronavirus, however, is not able to read this information. So in order to replicate itself, invades our cells, transmits its RNA, and synthesizes its components using host cell mechanisms. For millions of years, this is how the species survives and evolves. Four characteristics are essential for making a virus dangerous and capable of inducing a pandemic. Coronavirus biology comprises all four characteristics. Coronavirus has increased transmission potential compared to other respiratory viruses. COVID-19 creates many asymptomatic carriers which continue to spread the virus. There is a long incubation period between infection and symptoms onset, during which people are asymptomatic but contagious. It may, although rarely, cause deadly complications. The molecular diagnosis of COVID-19 is based on the detection of viral RNA in samples. However, a sample taken from a patient will contain a mixture of viral and human genetic material. Moreover, viral RNAs in minuscule quantities cannot be measured. Using an appropriate enzyme called polymerase, we amplify a specific segment of viral RNA after we have reversibly transcribed it to DNA. This reaction of polymerase is extremely specific for the viral genetic material and does not affect human DNA. It is also continuous, comprising several similar cycles like a chain. Polymerase chain reaction ends up producing millions or billions of copies of this specific viral genetic segment, which can now be measured. Coronavirus transmission is prevented by hand washing and by using protective masks among patients and healthy individuals. The virus is transmitted by touching infected surfaces and then our eyes, nose, or mouth. As a respiratory virus, if appropriate distance and the use of masks are not applied, then droplets of cough or sneezing containing viral particles may infect other people. The virus first infects in the interior of our nose, pharynx, or bronchi. There it hooks and invades resident epithelial cells. Its envelope dissolves and genetic material is released inside our cells. From that moment, the virus takes over the cell replication machinery. Our unsuspected cells will follow the virus instructions. The instructions are clear: replication of thousands of viral RNA copies, production of its ingredients, and finally cell self-destruction. These genetic copies, together with the fresh viral proteins, will synthesize thousands new viruses. They have one purpose only: to replicate by infecting new human cells. Our defense system stands up immediately in order to cancel virus plans. A battle is about to begin. On the blue corner, a creature bearing effective strategies to defeat advanced immune systems encountered during the millions of years of its existence, coronavirus. And on the red corner, the most sophisticated and evolutionary adapted defense system in nature, our immune system. It will defend our health at any cost, sometimes risking its own integrity and existence. Our body will respond promptly. On D-Day, non-specific immune cells will be sent at the area of infection. They consume viruses and infected cells in order to slow the progression of viral invasion, precluding its fast spreading. We know that in a few days from now, a specific and targeted counterattack is about to be unleashed. But when the virus enters our cells, it becomes invisible to immune cells. However, our defense system has evolved to overcome this behavior. First, virus cells invasion triggers host cells to produce and secrete warning signals in the adjacent area, the interferons. Neighboring cells receive these messages and strengthen the resistance to the upcoming virus attack. Second, infected cells display virus particles called antigens in their membrane. Exposed antigens will attract cytotoxic lymphocytes in the area. Cytotoxic lymphocytes will recognize virus antigens and will destroy viruses and the infected cells. But coronavirus cannot be defeated so easily. It unfolds its own tricks. Coronavirus vanguards will secure the area to facilitate the spreading of the viruses that follow. An operation shield will be deployed to block the transmission of warning signals of interference to our neighboring cells. They will face the upcoming coronavirus attack in a few minutes unprepared. Furthermore, coronavirus prevents its antigen presentation on the surface of infected cells, thus precluding its recognition from the patrolling cytotoxic lymphocytes. Meanwhile, the virus is replicating and spreading fast. A solution should be found immediately. It is time for natural killer cells to take action. As they scan the area, they are able to sense the stress of infected cells and feel the absence of specific host proteins in their surface. Without any delay, natural killer cells will efficiently destroy any contaminated cell. If the primary steps of our defense work efficiently, our body will unleash its final attack. Upon viral invasion, specially trained dendritic cells arrive in the affected tissues. They not just consume viruses. Proud of their achievement, they present modified pieces of coronavirus to other cells. Moreover, blood monocytes are attracted from signals emitted from the area of infection. They differentiate into macrophages and exhibit a similar behavior. The mission assigned to these cells is to steal and process important secrets concerning structural characteristics of the virus components and then to present this information to cells of interest. They are called antigen-presenting cells. This secret information will be delivered and will upgrade cytotoxic lymphocytes, thus improving their ability to kill viruses and triggering the formation of memory T lymphocytes. Significantly, specific helper T lymphocytes recognize the importance of the stolen enemy secrets and transfer this information to beta lymphocytes. At the same time, they regulate the final tuning of the defense systems of the second phase. Once they receive and process this information, beta cells transform into plasma cells with subsequent massive production of specific antiviral weapons, the antibodies. Antibodies are synthesized based on the key information regarding virus structure, representing a precise and targeted antiviral defense system. Once they attach to the viral surface, they destroy viruses boosted by the activation of the complement system. They prevent virus entry into our cells. They induce the formation of non-functional virus aggregates, which are an easy prey for our scavenger cells, the macrophages. If everything goes well, which is the typical scenario in most of the cases, coronavirus infection is limited to the upper respiratory system, and the virus is eliminated, causing mild or no symptoms at all. However, even in cases of a mild course of the infection, we may remain contagious to the people around us for 1 to 2 weeks. If coronavirus is not promptly eliminated, the battle will be transported deeper in our respiratory system, in the lungs. Things are getting difficult now. The virus has escaped the first line of defense, and the battlefield is thousands times bigger. Our lungs' tubular system is very extensive, ending in small air sacs known as alveoli, where blood gets oxygenated. Both human lungs contain more than 500 million alveoli, with a total surface covering 100 square meters, similar to half of a tennis court. Millions of coronaviruses arriving in the area, together with thousands of immune cells in a battle to keep the lungs clear. As a result, several toxic substances and cytokines are released, causing local or more extensive lung inflammation called pneumonia. Pneumonia causes high fever, shortness of breath, and exhaustion. More often, the outcome of pneumonia is successful and the virus is finally eradicated. However, exhaustion and related symptoms may persist for several weeks until the injured lungs recover completely. In a few cases, the coronavirus causes severe lung inflammation, microthrombosis, and extensive alveolar damage, leading to acute respiratory failure called acute respiratory distress syndrome, or ARDS. Blood oxygenation is compromised and the patient may need mechanical breathing support through a ventilator. As we have seen, if our first defense barrier is strong enough, it will block or limit coronavirus invasion efforts and will give us enough time for the preparation and release of our second phase of counterattack with more precise and targeted elements, such as upgraded cytotoxic lymphocytes and specific antibodies, resulting in final virus elimination. But if the coronavirus plans succeed, it will face a weak immune resistance upon its initial attempt for invasion. Escaping from our immune traps, the rate of virus replication will become uncontrolled. Suddenly, our immune system wakes up from sleep. In an effort to compensate for its previous negligence, our defense mechanisms abolish any tuning of the immune response, thus risking an uncontrolled immune reaction. Coronavirus will affect most of our organs and tissues, causing acute respiratory distress, thrombosis in small and larger vessels, renal insufficiency, and multiple organ failure. Our entire body is now a battlefield. Advanced stage, several comorbidities or other yet unknown factors may contribute to such outcomes. Without any control now, our immune cells panic. Strange signals, toxic substances, and cytokines are released everywhere in our body. In this stage, the virus itself, as well as our immune cells and the toxins released, cause extensive injuries in several tissues, creating a deadly feedback and chaos. The situation is getting critical. Our exhausted immune system becomes an easy prey for lurking pathogens, leading to sepsis and total body collapse. Our immune system retains every piece of information derived from its contact with coronavirus. As we have seen, the results of this interaction is the generation of memory T lymphocytes and the production of specific antibodies, lasting long enough, ready to defeat any future attempt of coronavirus invasion. We know more than 200 years now that a similar immune response would emerge if our immune system is exposed to inactivated or live attenuated viruses through vaccination. Indeed, some of coronavirus vaccines available today use inactivated or live attenuated coronaviruses as antigenic stimulus. This type of vaccines induce an immune reaction leading to the production of memory T cells and specific antibodies. A different type of vaccines is based on the synthesis of specific protein particles of coronavirus, such as the spikes. Synthetic spikes are joined together in self-assembly platforms or on virus-like particles and administered as antigenic stimuli. They are called nanoparticles since their size is very small. It has been demonstrated that this type of vaccines elicits similar immune response with production of memory T cells and specific antibodies. In order to create a genetic vaccine, we have to analyze the genetic code of the coronavirus, to detect the RNA segment responsible for the production of its spikes, to synthesize a similar mRNA particle, and to use this synthetic mRNA particle as a vaccine. However, free RNA molecules are highly unstable in our body. They will immediately undergo degradation before they manage to activate our immune cells. An idea was to encapsulate and protect mRNA molecules into spherical lipid nanoparticles, which are then used as delivery vehicles. Lipid nanoparticles containing coronavirus mRNA are recognized by immune cells. Once they enter a cell, lipid carriers dissolve and mRNA synthesizes coronavirus spikes without entering the cell nucleus. Spikes represent the antigens which will trigger our immune response to coronavirus, generating memory T lymphocytes and antibodies. During this process, lipid nanoparticles induce an inflammatory response, causing pain at the injection site, tiredness, headache, and fever for 1 to 2 days. Adenoviruses are common viruses that cause mild respiratory infections in humans and other mammals such as primates. Adenoviruses can be genetically modified to reduce their virulence and then be used as gene carriers between species. How can we transform an infectious adenovirus to a gene delivery system? We choose a common cold adenovirus, we remove all genes responsible for its replication and toxicity, and we insert the relevant gene of interest in the free space created. In this case, we insert the coronavirus gene responsible for the production of its spikes. However, adenoviruses contain DNA, while the piece of genetic material of coronavirus is RNA, so it cannot be attached. For this reason, we have to synthesize a DNA fragment complementary to coronavirus RNA. Now our synthetic cDNA contains information for the synthesis of coronavirus spikes and can easily be inserted to adenovirus DNA. From that moment, this replication defective adenovirus can be used as a viral vector vaccine. Following vaccination, adenovirus vehicle enters our cells, delivers its genetic material, including coronavirus spike gene, and then dissolves. But spike gene is a piece of DNA and it cannot synthesize spike proteins unless it enters the nucleus, transcribes to mRNA, travels back to cytoplasm, and produces coronavirus spikes similar to mRNA-based vaccines. The synthesized spikes serve as antigens for immune activation. Currently available viral vector vaccines use a replication defective adenovirus of the chimpanzee common cold, specially engineered to carry the coronavirus spike gene, and 2 modified adenoviruses of the human common cold, specially engineered to carry the coronavirus spike gene, the serotypes 26 and 5. As we witness an ongoing extensive research for novel therapies against coronavirus, the first virus mutations emerge. Every scientist on the planet knows that this phenomenon was highly expected. Mutations are spontaneous modifications in the genetic material of all living organisms in the process of evolution. And coronavirus is not an exception. Most of coronavirus mutations do not hold any functional significance. While other mutations reduce its ability to attach to our cells, these strains are defective and will disappear. However, there are mutations that enhance coronavirus' ability to bind to our cells, thus increasing its transmissibility. These advantageous strains will finally dominate over all previous phenotypic variations. Coronavirus is not going to win the battle with humans. Neither will it disappear, however. Reaching a balance, it will find its place in our ecosystem as another pathogenic virus like many others. With a total mortality of 0.5 to 1% or 5% in the very elderly, COVID-19 is not an extremely deadly disease. However, it is very contagious, affecting simultaneously large parts of the population, resulting in massive hospitalizations and intensive care unit admissions, and pushing national health systems to their limits. For this reason, the scientific community has responded promptly. Let us hope that we will witness similar scientific responses to other issues as well, such as global poverty, climate change, deforestation, massive forced migrations, wildlife protection, or improvement of global health, thus making humans less morbid and vulnerable. We have to remember that none of these issues is irrelevant with the risk of a pandemic outbreak in the near future.